Kelly.
Are there other species of native snails that these are like out competing? Like, is there a concern, competition with native species?
There's some studies that show that they negatively impact the foraging behavior of a couple of different native species. I don't know about here exactly. They've been here for a while, but I think it's boating. Yeah, they're pretty small.
So we do a lot of water sampling for the toxins from harmful algal blooms. And we also did a study looking at zebra and quagga mussels to see if they're kind of sequestering some of those toxins. I think there's potential that the snails might also, because some of those are spots that you're sampling are hot spots, like once they get a harmful algal bloom, it's chronic for the rest of the summer.
Ooh, that's a good question. I might call on Simpson and Newtile, because they're the toxicologists and the physiologists. So, do you guys have any insights on sequestering?
Bioaccumulation?
Yeah.
Yeah.
I mean, we have a couple students that have looked at metal accumulation in the mystery snails, and they do accumulate pretty high levels of lead, zinc, and
Copper.
Yeah, copper. I don't know about organic snow. We
Okay.
haven't really, just because the analysis is a lot harder.
Mhm.
But we do know that they harbor quite a bit of metal contaminants.
If you have methods for analyzing that, that'd be interesting. We have plenty of tissue.
I was gonna say, it might be worthwhile, because I never thought of snails, I was just thinking of,
Yeah.
bivalves off the get-go, but the results came back and they were really interesting, so there was interest in continuing.
looking at that, but I think this would be another really
Yeah.
good organism.
Yeah, certainly.
Okay.
Certainly.
Thank you.
Yeah.
So what would you tell a member of the community if they asked you, if I find one of these things, what should I do with it? First, what can I do with it? Like, would I be violating any laws if I found a bunch of them, took them home and ate them?
No, you would not.
So what would you encourage me to do? Because I'd be eating your study organism.
Well, you shouldn't be doing that. The parasites that they harbor alone is frightening. Like if you look at the foot when it's up against glass, you can see all the little things encysted in it. Oh, it's like Starry Night, man. What I would recommend that you do if you find them, the best thing you can do is obviously report them, because that lets us know that they're there. You can also give them to us to have in freeze, euthanize, all that stuff. If you're in the middle of nowhere, I know a lot of people tend to huck them into the woods. Not that that's necessarily your go-to. The best thing is to report it, let people know, and then
Remove them?
remove them. Yeah, but you can buy them on Amazon.
Or aquaria or
Pond algal control. Yeah, yeah, and there's nothing stopping Amazon from selling them and shipping them. So, yeah.
Yesterday, there was a comment in in the presentations about the possibility of river otters being on the park. Did you consider the possibility that your predator in Thompson's Bay could be a river otter?
I didn't hear that, but I don't. Kelly says no.
I think they're probably raccoons that are eating them.
Raccoons? What makes you think raccoon versus otter?
Because raccoons have really textile hands, and otters will eat smaller organisms. They will sometimes eat mussels, but really they prefer bigger species, small fish and crayfish.
Okay.
There's a lot of meat in one of these mystery snails, though.
Yeah, it could be.
Yeah, it could be worth the
I mean, I would love to set up like plasticine ones with a camera next to it or something, just to see who's chomping on it. But yeah,
I was going to say our mussels that were also in Thompson's were smashed the same way.
Yeah.
That was, yeah.
yeah. So something's smashing
Yeah,
in Thompson
something's
Bay.
breaking them, yeah.
Okay. All right. I think I'm out of time.
Thank you.
Thank you, guys.
All right.
All right, we have more snail work coming up here. So Dr. Adam Simpson from Barron is going to talk with us about the genetic side of things here with mystery snails in this part of the state. All right, I'll hand it over to you. Here's the just left and right arrows.
All right, thank you, Casey. So I am presenting some research performed by some excellent students who have moved on to greener pastures, Aaron Seymour and Phoebe Will, as well as my collaborators at Barron. And we were sort of doing this project as an extension of all the demographic and surveying we were doing out in Presque Isle, as well as some of the inland water bodies in western Pennsylvania. So it's obviously no surprise to anyone that invasive species and introduced species can cause problems for the habitats that they potentially invade. I'm highlighting ones that everyone probably knows about, but there's a reason for that. These are very charismatic invasive species that almost anyone, if you stop someone on a boat or they're fishing off a pier, they know what a zebra mussel is, for example. And they could probably tell you what they do. There's impacts on ecosystems, which maybe some people don't care as much about, but things like mussels, native mussels that are an extremely threatened taxonomic group. As Andrew's talking about, these things are often quite large. Many species are large compared to zebra mussels. The zebra mussels can prevent them from feeding in the way they usually do, which can be devastating for them. There's outcompeting for resources. And zebra mussels can affect humans too. These are probably the reasons that the lay public is concerned about. What is it doing to pipes? What is it doing to your boat motor? So these guys can reach massive densities that can be a major problem, at least locally. Other very charismatic invasive species, most people can point to a round goby, which is native to same area as zebra mussels, Eurasia. These guys can reach high densities. If you've been fishing off of State Street or South Pier, the bottom is just carpeted with round goby. They're delicious, but unfortunately, they will eat pretty much anything. They do eat zebra mussels, which is kind of nice, but they also eat other things This is a somewhat nebulous group of ovoviviparous snails. In Lake Erie, they arrived in the early 1900s. They sort of made their way east from Lake Superior over. The first detection in Lake Erie was in the Sandusky region in the 1960s. I think it was 1968. And they finally made their way up, they probably here before then, to actual Erie County, up by us in 2006. They've likely been there for much longer than that, but this is the first time they've been reported. So it may not seem like, at least for most of us, 2006 doesn't seem like it was that long ago, but it was almost 20 years ago. So they've been here a while, and if you talk to anyone, and we've asked people as we're sampling in our kayaks along the shore, have you seen any giant snails? They're like, oh yeah, we see these all the time. And they don't associate them with harm. They don't associate them with invasive species. So we often, for introduced species, we assume that if they're here in high densities, they didn't originate here, they're probably impacting the ecosystem in some way. The general assumption is that they're going to be outcompeting other native species that are occupying the same niche. We have a lot of native freshwater snails. Most of them are quite a bit smaller than our mystery snails are. And we have some evidence in our research group for accumulation of inorganic toxicants, so things like heavy metals, lead, zinc, copper, that can accumulate in their shell, which we're concerned if other animals eat them that could lead to bioconcentration or bioaccumulation of different trophic levels. And we've seen some evidence, unfortunately, suggesting that you probably shouldn't eat these snails. There's a lot of parasites in them. If you put these guys in a cup of water, a lot of these parasites, their life cycle, they'll just sort of burst out, start swimming around. Most of the ones we've seen have been trematode parasites, so flukes. They're kind of gross, and you would assume with bigger body size, they can accumulate more parasites. So it's not necessarily that they're causing devastating harm to these ecosystems, but they're not necessarily helping either. So, unfortunately, we don't really know much about mystery snails, and perhaps that's why the name is appropriate. We don't necessarily know what they're doing to these ecosystems, but we also don't necessarily know what they are. That's kind of the major concern we're running into. Dr. Beatty mentioned our two morphotypes here. These are the two species that have been described as being present in Lake Erie and in Erie County in western Pennsylvania, are Chinese mystery snail and Japanese mystery snail. which until relatively recently were categorized in the same genus, just different species. Both are native to Eastern Asia. You'll notice there are some differences in shell morphology, although it's not consistent. We do see a lot of plasticity in shell shape, even coloration across different sites, despite them being the same species. So the question here, before you really move forward with this type of research, is understanding how different they are. And does it really matter? You know, if they have similar demographics, similar ecology, does the management practice, the strategy for controlling them, does it matter? Do we need to create separate management plans for these species? So, we've done some previous research. Dr. Beatty gave more details about this, but last summer, we identified 2 different morphotypes that were present across 5 sites in western Pennsylvania. The Chinese mystery snail morph was located or identified in all 5 locations, and we found the Japanese mystery snail in Pymatuning and Lake Arthur. So there were some instances where we had overlap in distribution of the Chinese and Japanese mystery snails. So our plan moving forward, and this changed for reasons I'll explain shortly, was to understand population genetics. That can give us some insight as far as estimates of population size, genetic variability. We can get some information about invasion routes, how they've spread throughout the state. And there's a lot of great ways to do this. I usually use microsatellite population genetic analyses, which is a great and robust technique. However, if we're not necessarily sure what we're working with, it's not really appropriate to do these types of analyses. So this project started as just a little bookkeeping mini project just to make sure we knew what we were working with. So you got to kind of know what you're working with before you can move forward. So the main objective of this project was to just make sure we've got Chinese and Japanese mystery snails that we've collected. So we took a subsample of the individuals that we collected from the 5 sites, or the 5 locations, I should say. We did, as Dr. Beatty showed you, sample multiple sites within those water bodies. You can see that depicted here. And we just selected 10 random snails from each of those sites within each water body. As far as identifying the snails, we used genetic barcoding, which is a pretty standard technique. We isolated DNA from our snails through a variety of different tissues. Some tissues work better than others. I don't know if any of you guys have done snail genetics, but it's kind of a pain. Their DNA is not very clean, so it can be kind of tricky. We isolated DNA from the foot of the snails as well as their faces. Just kind of, yeah, I came in one day and Dr. Newtile had just filled my freezer with tubes of snail faces. But it's a good source of DNA. So we extracted that DNA, we did your typical tissue digestion, we purified DNA, and we quantified and to some extent qualified it to make sure we had good samples to send out. Next step was PCR, and this is how we amplify specific regions within the genome that we can use to identify different snails. A common method for invertebrates is to use the mitochondrial genome. So we have a particular gene here called the cytochrome C oxidase, or CO1 gene. That's extremely popular. It's used as a universal barcode for many invertebrates. It's pretty reliable. The mitochondrial genome is very small compared to the nuclear genome, so we'll get to that. Some potential issues here. But how this process works, I mean, most of you guys know how PCR works. We amplify particular regions of the genome that we extracted, and what that's going to create are barcodes. And the set of primers I used are the Fulmer primers, which are the most commonly used CO1 primers. So this created or amplified a 658 base pair region. We had a bunch of copies of it, and that allowed us to sequence them. And what you would expect if we have different species, that section of 658 base pairs is going to be slightly different. And we expect similarities within species and differences between species. So, what you typically do to make sure that things work before you send it out, you run gel electrophoresis just to look at bands. All these bands should be the same. Unfortunately, as any of you genetics folks know, sometimes things just don't work. Even if you've done it 100 times, the 101st time it just doesn't work. So this was a problem, and we discovered most of this through over spring break, and it was really terrible. And we decided to reevaluate our method, and in addition to adding all the normal stuff for PCR you usually do, we added a little pizzazz. On Fridays we have Jimmy Buffett Fridays. and we just play all Jimmy Buffett songs. And the students noticed that when they played Margaritaville, the PCR worked. So we went from like nothing, I'm telling you, same method, same samples, excellent bands, right? They're all like the same size, it's perfect. So we stuck with it, and from here on out, every single PCR cycle, they played Margaritaville on loop, and they kept a counter going, and they made it over 100, just 100 consecutive Margaritavilles, and they were loving it. We wear leis and everything. So it worked. So what you do, send those samples out. We now have amplicons. We have amplified regions of those barcodes. We send them out for sequencing. You basically just perform the same reaction again. So you break apart those amplified regions and redo it. But this time we have fluorescent labeled or tagged nucleotides that allow us to actually figure out what the base pairs are as that reaction continues once again. So this time we're doing these things separately. So what you're going to get ultimately are a forward barcode and a reverse barcode. And if you kind of flip them around and try and align them together, you can get a better idea of what you're actually looking at. And sometimes, with a forward or reverse barcode, you get some missing base pairs, and this allows you to kind of fill in the gaps and make sure you've got a nice solid barcode that you can use for your next step. So after the alignment of our amplicons here, we were able to compare them to other species. So in the NCBI nucleotide blast database, this allows you to just compare your sequences with those that are out in literature, look for similarities, and that can give you some idea of genetic identity. So we get a percent match that is somewhat variable sometimes, but it gives us a decent idea of what we're working with. So what we found. Cool, we found Chinese mystery snails, we found Japanese mystery snails. Unfortunately, or maybe fortunately, I don't know. We also had positive IDs for 2 other species that have not been reported in this area, or in the Great Lakes region for that matter. One of them shares the same genus. The other one, Margaria melanoides, is very, very different, and we'll get to that shortly. But one thing to point out, the 3 species up top, they all look like Chinese mystery snails. We've noticed some slight differences. I'm not sure we associated them necessarily with different species, but we see interesting patterns of dimpling on the shell, as well as some little spiky ridges that go around the outside. But they all generally look quite the same. The Japanese mystery snails are relatively distinct, at least within certain sites that we look at. So we've got 2 little morphotypes here, and it's complicated the situation because now we potentially have 4 species where we thought there were 2. So this is the color code I'm going to use here. The snails are obviously not those colors. Those are not necessarily pictures of their shells, but at least shows you where we found each of these species that we identified here. Press Isle, we identified 2 different species. Interestingly, C. chinensis was not one of them. The assumption was that most of the snails here are C. chinensis. None of them are, according to our data so far. We did find Margaria melanoides and C. cathiensis at multiple locations, the same ones Dr. Beatty talked about. So we've got Feather Platform, Lily Pond, Graveyard Pond, and Thompson Bay. In Lake Canadota, we only found the Margaria melanoides species. In Lake Arthur, so in Moraine State Park, we identified 2 different species. The Chinese morphs were all Margaria melanoides, and we had some C. japonica as well. In Lake Pleasant, interestingly, this is a very small water body. We found 3 different Chinese mystery snail morphs that were located within that tiny water body. And for Pymatooning, we found all 4. And there were some sites that I think is kind of cool that had multiple species of Chinese mystery snail morphs as well as Japanese mystery snails. So this is really confusing. It's kind of a mess. And there's a reason for that. This is not news that snail genetics, phylogeny, taxonomy is confusing. People have made careers out of just sort of reconfiguring and trying to find evolutionary and genetic relationships between these species. They all fall relatively neatly within one family of ovoviviparous snails, but unfortunately, as far as genus, there are a lot of different genera here, and they all look very similar. So this is what we'd consider a cryptic species complex. The names have changed, the players maybe not so much. So I know the Sapengopalladina japonica has changed its genus, it's heterogen now, but this stuff changes depending on what is, and the complication that arises from trying to identify them genetically suggests that hybridization could occur. I don't know if that's been documented in the US, but it certainly has in their native range. We do see some crossbreeding here, which can really complicate our definition of what a species is, what a genus is. So, some uncertainties here that I should point out, because these data are somewhat preliminary. In our Chinese mystery snail morphs, again, they all look very similar, but genetically they are different. Our Margaria melanoides, this is what they're supposed to look like in China. This is a relatively rare species. They look like mystery snails. They have an operculum. They have relatively tall spires, but you notice that there's all these really cool ridges and spikes, and we haven't found anything like that here. So that's potentially confusing and confounding. There's a couple different explanations for that. One is that the genetic markers we use are just not sufficient to truly tell if this is Margaria melanoides. So we need a bit more genetic material or genetic information to really figure that out. The other explanation could be they've been here long enough and they've undergone either through drift or local adaptation. We've just seen differences in shell shape that have changed over time. So they more closely resemble some of the other species that have already been here. As far as percent ID, so when you do these comparisons, the NCBI BLAST, you do get a percent ID based on the information, the genomic information that's previously published and publicly available. For our Chinese mystery snail morphs, that percent ID was a bit lower than we saw for Japanese mystery snails. Very, very confident that the individuals we identified as Japanese mystery snails are in fact Japanese mystery snails. These guys, unclear. I do believe that we have more species here than we think we do. I can't, gun to my head, I wouldn't necessarily say we have 3 different species, but there's something funny going on. So how can we fix this problem? Well, we can use different barcodes. We use the most common one. CO1 is very popular for genetic barcoding, but there's other things we can do. There's other places we can find barcodes and test those out. So is this a big deal? Should we be concerned with this? Does this add an extra layer to management efforts? If you're working for Sea Grant or you're DCNR, whatever it happens to be, it could be that managing 2 species is a lot easier than managing 4 species. But what could complicate this, or what could make this simpler, I suppose, is if they don't really differ much ecologically. If they live in similar places, if their abundance is relatively similar, if their reproduction is relatively similar lifespan, the management efforts might not need to change. But it does potentially bring up a concern as far as how we manage the population, how we explain to the public that we have multiple species. So what are we doing going forward? Like I said, we don't have much genetic information that's published out there. There's mitochondrial genomes that have been published, but we don't really have much for the rest of the genome, which is the vast majority of it. So we are currently working on whole genome sequencing with long reads, which makes it a little bit easier to assemble those genomes and figure out what's going on. We can also use that to mine for microsatellites and other genetic markers. Once we have that information, we can perform microsatellite analyses to look at differences between populations, look at population, genetic variation, and so on. And ultimately, we need to try and determine if they differ at all. Do they behave differently? Do they eat more or less food? Do they live in slightly different places? This is information that's needed to truly figure out what we need to do moving forward. And we will continue sampling, and hopefully we'll share some updates next year. So I'd like to thank all of my collaborators and helpers, the former Snail Squad samplers, Matt Gruhl, and the other Mike Campbell for helping me out with some genetics advice, and of course Jimmy Buffett for helping our PCR work. And with that, I will take questions.
All right, we can do one quick question.
I do have a question. So the one snail that you showed what it looks like in Asia and what it looks like here, or, very different.
Yeah.
What about the other species? I'm just thinking how long have they been here, evolutionary pressures, like if it changed, do the other Chinese morphs look like they do over in Asia right now? Do they look more similar?
Yeah, so the Cathiensis looks almost identical to chinensis. So those two, it makes sense that we might get those confused, and it's possible that the initial reports of we have Chinese mystery snails here, it might have been Cathiensis to begin with. The lowest detection, the least detected species was chinensis, actually. Cathiensis and Margaria was everywhere. So those look relatively similar to what they look like in Asia, but Margaria, I don't know. Again, that could be either mis-ID that we need to keep looking at, or it'd be really cool if it was some sort of local adaptation, but obviously it'd be better for us if it was not. Just another subspecies, but.
Great, thank you.
Thanks.
All right, so the next two presentations we have are undergraduate students, so if you are judging and have a judging packet, you can pull those out now. The first one is Olivia Hodgson. She's coming up now from Penn State Barron. And she's looking at polychlorinated biphenyl concentrations in Lake Erie fishes. And then you can use this to advance your slides if you want. It's just the right here. Right.
Hello everyone, I am Olivia. I'm actually a senior this year at Penn State Barron. And thank you for having me present on this beautiful morning. I've been working on this project for almost 2 years now with Doctors Nutile and Simpson, as well as a few other individuals. We investigated the concentration of polychlorinated biphenyls in the tissues of Lake Erie fishes, as well as explored their possible human health impacts. So what are PCBs? As their name suggests, polychlorinated biphenyls are two joined phenyl rings. So they are two 6-carbon rings joined together at the center, and they vary greatly with respect to their number and possible arrangement of attached chlorine atoms. And there exists 209 different congeners. PCBs were very popular in manufacturing and industry as refrigerants, lubricants, and very effective flame retardants, but they were banned by the Environmental Protection Agency in the 1970s due to their possible negative ecosystem and human health impacts. And all 209 congeners of PCBs are hydrophobic. So whenever they are exposed to an aquatic habitat, they are not going to hang out and float around in the water. Instead, they are going to sequester themselves in the sediments at the bottom of these habitats. So because they are these two joined phenyl rings, PCBs have a very high chemical stability, and therefore they will persist in aquatic habitats because they don't break down naturally through natural processes. And because they are hydrophobic, our aquatic invertebrates can be exposed in the sediment, and through consumption, they can bioaccumulate throughout the trophic levels and possibly be exposed to humans. We don't have to go very far to study this. Lake Erie is very nearby. It is a famous lake with many attractions, and it has witnessed a lot of human activity along its shoreline. So we have major US cities like Buffalo, New York, Erie, Pennsylvania, Cleveland and Toledo, Ohio, as well as Detroit, Michigan, upstream there. And so the shoreline of Lake Erie has been very busy with a lot of industry and manufacturing, and therefore exposed to a lot of PCB contamination. Lake Erie is also a very popular recreational fishery, and it witnesses both local residents and a lot of tourism of people who come to fish these waters. There is currently a consumption advisory on fish consumed out of Lake Erie due to PCB contamination. That consumption advisory is one meal, which PADCNR considers to be an 8-ounce fillet per month. And that brings up a very important research question for us. Are these consumption advisories actually protective of human health? And that brings us to the first research objective, which was to quantify the exact concentration of PCBs within the tissues of Lake Erie fishes. The first step was to go fishing. So we went out fishing and we caught 5 different species of fish: walleye, steelhead trout, bluegill, yellow perch, and freshwater drum. Once these fish were caught, they were skinned and filleted to represent what an individual might consume as a meal. For each of these 5 species, there were 12 replicates of each. After they were filleted, they were then ground and homogenized, and they went through a sonication process to release the PCBs from the tissue. The PCBs were then extracted from the tissue using an organic liquid-liquid extraction process. The tissue was further broken down through an acid cleanup process using sulfuric acid. Sulfuric acid is colorless, but once it begins to interact with tissue, it turns into various shades of orange, as you can see in this picture. The remaining concentrate was then stored at 1 milliliter in these GC vials, and then they were analyzed via gas chromatography mass spectrometry. So each PCB has a very unique footprint on the mass spectrum. And the first objective, I guess, that we had to do was qualify the PCBs. So we had to be able to make sure that we could detect PCBs at any degree, which we were able to do. We then quantified the concentration of PCBs to make sure that we could get that exact concentration in tissues. And as I said before, there are 209 congeners of PCBs. That's a lot, so we used a representative sample of 27 congeners. This first figure is what we found, I guess, from the GCMS, and this is a total concentration of PCBs that we found in fish tissue. And the first thing that I want to point out is that there's a lot of variability in these box and whisker plots. And that is not surprising, because fish don't stay still in their habitat. They are going to be moving around looking for different organisms to feed on, so it's not surprising that individuals within the same species are going to be exposed to a different degree of PCBs. This next figure is that same data, but we're going to look at it a little bit differently and compare it to the consumption advisory that is in place from the PADCNR. Everything that falls below this green line here is safe to eat generally because there is not a concentration high enough to necessitate a consumption advisory. Everything that falls above the green line and below the yellow line is safe to eat at 1 meal per week, and everything that falls or comes above that yellow line is within the threshold of 1 meal per month. For the purposes of this presentation, we don't need to worry about anything greater than that because we did not detect concentrations that high. So why do we care? Why did we care? Why did we do